Hi, my name's Helen Messier, and I'm the CMO of Viome. And I'd like to share with you the power of precision wellness and how we can use that for truly personalized nutrition. First of all, I'd like to introduce you to Joe. Now, Joe is suffering from multiple chronic conditions. And unfortunately, as we all know, there are far too many people like Joe around. In fact, chronic conditions account for more than 60% of deaths worldwide, and more than 50% of people are currently living with at least one chronic disease, and the incidence just keeps increasing. But Joe doesn't want to be one of those statistics anymore. He really wants to get healthy, and he knows that diet is an important part of achieving this. But where does he start? There are so many different types of diets out there. Should he follow the USDA food pyramid, the Mediterranean diet, the ketogenic diet, the paleo diet? And he reads a lot of articles, and the popular press just isn't much help. It keeps contradicting itself. He's even gone to the literature and found a study that shows a low carb diet was better than a conventional low fat diet for weight loss and for some heart disease risk factors. And he also found a website that listed a comparison of diet studies. And in most cases, the low carb diet seemed to win out over the low fat diet for weight loss. But in this one, it showed that even when people lost weight on the low carb diet, instead of losing stored fat, the body was mostly burning lean muscle mass and protein. Now he's getting really confused. How does he know what the right diet will be for him? The popular media and even the scientific literature seems to be contradicting itself. The evidence is not all that clear cut. But because the majority of evidence does seem to suggest it, and because it's really trendy right now, Joe decides to try a paleo diet. Now, unfortunately, 3 months later, even though he's lost some weight, his cardiovascular risk factors have increased, and he's starting to experience some mild chest pains. This is confusing again. What's going on? The paleo diet did work for a lot of people, and Joe can't figure out why he's so different. Then he saw this article. A new study links L-carnitine in red meat to heart disease. Uh-oh. And he found the actual study showing that intestinal microbiota metabolism of carnitine produces trimethylamine, which is further metabolized in the liver to trimethylamine N oxide, or TMAO, which accelerates atherosclerosis, or hardening of the arteries. Wow, it does confirm the importance of our microbiome, but which species associate with TMAO production, and does Joe have these present in his microbiome? Here's another look of how that happens. The food the food that we eat, the carnitine and the choline, we ingest it, it gets metabolized by our gut microbes, and it gets absorbed into our body to the liver. It gets produced into TMAO, and that leads to atherosclerosis. Well, we're making some progress, right? Now, at least Joe knows why he may have gotten worse on the paleo diet, and he learned a really important lesson that everyone is different. Up to now, we've been looking at the tip of the iceberg using epidemiological population studies, observational studies, and small randomized trials. But we really need to look at a person's molecular uniqueness to give truly personalized food recommendations. So let's dive in and take a look at how this molecular uniqueness is determined to produce precision wellness. First of all, I want to remind everyone that we are not really human, but we're a complex ecosystem of human and microbial cells and genes, all interacting with each other and our environment, especially our diet. Our body truly is mostly microbes. And the cover of The Economist, that microbes maketh man, is more true than we realize. Every surface of our body is covered inside and out with microbes, including a microbiome in our blood, which we had always previously thought was supposed to be sterile. The largest collection of microbes is in our gut, adding up to 5 pounds to our overall weight. And when we talk about the microbiome, we mean the collection of microbes in our gut in most cases. There are bacteria in the stomach, and as we go down the digestive tract, we find more and more bacteria, with the colon having the greatest number. Now, most of the bacteria in our gut are anaerobic, meaning they thrive without oxygen. And there are around 1.3 microbial cells to every human cell. And from a genetic perspective, we're outnumbered by microbial genes by at least 150 to 1. And our microbiome is not fixed, it's very dynamic, and it changes as we age and in response to our diet and our health status. Now, this is only a partial list of all the functions that we depend on our microbes for. They digest our food, they regulate our appetite, they control our metabolism, they produce essential vitamins and nutrients that we need. They make natural antibiotics and protect us from pathogens. They sustain the gastrointestinal tract and promote health and digestion that way. And they impact our mood and they orchestrate our immune system. Now, 70% of our immune cells live in our gut wall, and our microbes both train and control our immune system. And I really love this quote: the mammalian immune system, which seems to be designed to control microorganisms, is in fact controlled by microorganisms. So it's no wonder, with everything that our gut bacteria do, that they're connected to chronic disease. In fact, there's not one chronic disease that you can think of that has not been associated with a disruption of our microbiome at some level. And not only do our microbes influence our health, it's actually hard to think of something our body does that's not influenced, if not controlled, by our microbiome. Things like circadian rhythm. Our microbiome has its own biological clock that affects ours. Our mood, the ability to become pregnant and pregnancy outcomes. It affects our response to drugs, our ability to exercise at different levels, and as we said, our immune system is trained and controlled by our gut microbiome. And it's clearly connected to and influences our brain, as we've always intuitively known when we refer to gut feelings. So it's reasonable to propose that the composition of the microbiome and its activities are involved in most, if not all, of the biological processes that constitute human health and disease. So, what makes a healthy microbiome? How can we tell when it's been disrupted? And most importantly, what can we do about it? Well, we know that one of the most important factors that promotes a healthy microbiome is our diet, as the microbes are clearly the first ones exposed to everything that we take in. Ultimately, we're not just eating for our body's health, but also for the health of the microbiome, which in turn affects our health. So the first question to ask is, what is a healthy diet? And the answer is that there is no one healthy diet that's right for everyone. Now, some of us will respond positively to a specific nutrient, others won't respond at all, and still others will have a negative response. And our responses to foods can change over time. And most of this individual variability to food is determined by our unique microbiomes. Microbiomes as unique to us as our fingerprints. So, can we use the information found in our unique microbiomes to predict which diet will be most beneficial for people like Joe and for all of us? The answer is most definitely yes, but we need to see a number of things. We need to be able to see and identify all bacteria that are present, down to the strain level. Now, just to remind you of all the taxonomic classification system, it goes from the highest level of kingdom and then phylum, class, order, family, genus, species, and beyond species at an even higher resolution is strain. Now, strain level is important. This is just one example of why that is. In this case, of 91 bacteria that were assessed, only 5 strains were able to make GABA, which is a beneficial neurotransmitter, out of MSG. Now, whether you have these particular strains might determine whether you feel okay or really bad after eating MSG-laden foods. And in this case, it was a particular strain of Bifidobacterium longum called AH1206 that's been shown to colonize people's microbiomes when taken as a probiotic. Now, other strains of the same species have been shown not to colonize. And it's also been shown that certain bacterial strains could increase tight junction function. That's the things that hold the lining of our gut cells together. And it does that by modulating the protein components. And this ability is very strain specific. Now, whether the strain can affect tight junctions and the pathways that is utilized to do this. We also need to be able to see viruses, since many viruses are stable members of the human microbiome. They're very important because, unlike bacteria, they can readily cross the gut barrier and enter our bodies. We also need to see all of the fungi that are present because they also play a very previously unappreciated role in the microbiome. And the collection of fungi is called the microbiome. And of course, we need to see parasites if they're present as well. Now, just a quick reminder, we refer to 2 different cell types. Prokaryotic cells, those on the left, include bacteria and archaea, and they're cells that don't have a nucleus or any organelles such as mitochondria. And eukaryotic cells, those seen on the right, are all the other cells, including fungi and plants and even humans. These cells all have a nucleus and they have cellular organelles such as mitochondria. So to fully understand the microbiome and its effect on our health and how we can modify it with personalized diets, we really must see the prokaryotes to strain level, all of the viruses, and all of the eukaryotes. Now, science has been trying to look inside our microbiome for many years, but our tools have been limited. We started out using culture and trying to grow the bugs in petri dishes in order to identify them. Now, this worked for 20% of the microbes that could grow in oxygen, but 80%, as I said, are anaerobic, and they'll die as soon as they're out of our body. So we missed most of what is there. Other technologies have been able to utilize the DNA inside each bacteria in order to identify them. Now, these technologies, such as 16S ribosomal RNA gene analysis, sequences a small portion of the bacterial DNA. This is better than culture, but still can only identify bacteria to the genus level, not even species, and definitely not strain. And it will miss eukaryotes and viruses. Now, this is the technology that's still being used by most researchers and companies today. Now, when metagenomic or whole shotgun sequencing came along, it was much better. It can identify up to strain level and also see possible viruses and other eukaryotes. But because of low abundance, they're often missed. And it can also identify potential metabolic pathways that are present. And then there's metatranscriptomic sequencing or RNA sequencing, which again is a huge step better. It identifies all organisms, including bacteria, archaea, the yeast, mold, fungi, bacteriophages, parasites, and also RNA viruses. It can identify the living organisms which are active, so they're not confused by organisms that are dead, that are just passing through. And it can identify and quantify the activities of all the microorganisms. Now, this is a reminder to tell you that when genes which are found in DNA, are expressed, they produce RNA. So by looking at RNA, we see everything that's active downstream. DNA reflects a potential, and RNA reflects what is actually happening. Here's another way of thinking about it. All the cells in our body have the same DNA, so a genetic test cannot distinguish different cells. That's why you can do a DNA test from your blood or from the cheek swab or any other cell in your body. But every tissue expresses a different set of genes, leading them to perform different functions. RNA sequencing easily distinguishes different types of cells, so you can distinguish a liver from a kidney cell. So when we look at the microbiome, identifying a microbe with DNA sequencing tells us nothing about what they're actually doing. But RNA sequencing gives us a clear picture of what the microbes are consuming, what they're producing, and how they're interacting with each other. This is an analogy I like to use to explain the differences in these technologies. This is culture. It can identify the well-known ones that have a lot of air. 16S technology gives us a blurry silhouette of who's there. Metagenomic technology can identify who's there and what they're capable of doing. And metatranscriptomic technology, this is the technology that we use at Viome, can identify who's there and what they're really doing. With this technology, we can assess our clients' microbiomes and make personalized recommendations based on what their microbes are doing. It's what we call precision wellness. And we now have access to this transcriptomic technology at an unprecedented precision and low cost. With this RNA data, we can now identify the complex pathways and reactions that are being carried out by our microbes and by ourselves, and we can start to see what we need to do to optimize them. As you can imagine, this produces a lot of amazing data, and that's why we need a computer. So we use the power of artificial intelligence. With so much data, it's clear that the human brain, even the human microbial brain, just can't keep track of everything. So the power of artificial intelligence helps us understand what's going on in each person. We can start to answer these questions. How is your gut feeling? And how are your microbes doing? And what are your microbes doing? One of the things that we can see at a high level is that you might have bacteria that are known to produce butyrate. Why is this important? Well, because butyrate has many beneficial effects, including feeding the cells lining our gut to keep them healthy. It controls insulin sensitivity, neuronal health, it's an anti-inflammatory, and it even controls our own genetic expression. But just because the bacteria are present that have the potential to produce butyrate does not necessarily mean that they're doing what they're supposed to. How do your microbes actually make butyrate? Well, it can be made from 4 different substrates, resulting in 4 different pathways for butyrate synthesis. With RNA data, we can identify each of these pathways and see where butyrate might be coming from. And the important part is to identify the key enzymes that result in butyrate synthesis, those ones identified by the yellow highlights, to really know if your microbes are producing this beneficial short-chain fatty acid. And when we look at the thousands of samples that we've sequenced so far, we can see that not everyone's bacteria are really good at producing butyrate. The red dots are the people who have high levels of the key butyrate producing enzymes in their microbiome. The black is an average amount, and the green is people with very low levels of those enzymes. So if we don't have much butyrate production happening, what can you do to help? Well, you can increase it in a few ways. You can add in the bacteria that make it using probiotics or fermented foods. You can feed these bacteria with specific foods to turn on the expression of the butyrate production pathways. And you can even take butyrate as a supplement to make up for what your bugs are not producing. We can also see the presence of viruses. This is one example of viruses in the microbiome. So this is one of our clients. The top row is before a fecal microbiome transplant. As you can see, there's 99% of the activity is coming from bacteria. The second row down is after a fecal microbiome transplant, where now 44% are from bacteria, and the other activity in the microbiome is coming from viruses. And here you can see all of those viruses that are present, many of them being plant RNA viruses. So the majority of viral sequences in the human gut are actually coming from plant RNA viruses. The human gut acts as a vehicle for the dissemination of these viruses. And when we look at some of our preliminary findings, we can see that higher virus expression is associated with decreased overall physical health. And this is consistent with what's seen in the literature in that plant viruses can actually elicit an immune response and can be associated with symptoms. Some of the other things we can see is whether your microbiome is diverse. Now, we know that a healthy microbial human ecosystem is a diverse one. Just like a rainforest is healthier and more robust, the more biodiversity it has, it's the same with the microbiome. Biodiversity creates resilience, and the more diverse our microbiome is, the healthier it is, and the healthier we tend to be. And we can start to see the effects of our overuse of medications such as antibiotics. Unfortunately, many of our microbiomes are starting to look like this. But we can also see the incredible diversity of functions as the good guys and the bad guys try to reestablish themselves in a new ecosystem. And we can determine what foods and nutrients would be most crucial to ensure that the good guys don't lose the battle. We can also see whether your gut lining is being damaged by looking at the RNA in the cells that are being shed from the gut lining. As we already said, since the majority of your immune system lives in the lining of your gut, when this lining becomes damaged, the bacteria, especially the ones making LPS or flagella, which we can also see, can stimulate your immune system. And this can lead to chronic inflammation, which is really an underlying factor in all chronic disease. We can also identify when certain bacteria, usually the so-called bad guys, start forming gangs by turning on quorum sensing and biofilm production. And biofilms can damage the gut lining and make microbes resistant to antibiotics. So we can really see how food must be personalized. Food is really transformed our microbiome into beneficial or harmful products. And food feeds our microbiomes so it can promote the growth of good or bad organisms. So we can think of how people are responders or non-responders to specific foods. And this response is determined by their unique microbiomes. So let's go over some examples of how our microbes tell us what foods will be beneficial and harmful for each of us. In the world where the paleo is the in thing, we can see if you're eating too much protein and if it's not being digested properly. So if this isn't happening, the bacteria in the colon will ferment it and produce many harmful byproducts such as ammonia and cresol and putrescine and the TMA that we saw before. These harmful products can lead to inflammation, aging, and heart disease, among many other things. The key is that everyone needs different amounts. As we saw with Joe, his paleo diet was likely giving him too much protein and it was causing problems, where in other people it would be okay. So if we see someone is eating too much protein and it's harming them, we can recommend that they limit their protein intake. We can support protein digestion with things like digestive enzymes, and we can promote carbohydrate fermentation with probiotics and polyphenols, which all act to reduce protein fermentation. We can also see if someone is eating too much fructose from fruit and things like high fructose corn syrup. Fructose should really be metabolized in the small intestine. But if the small intestine gets overloaded, that fructose will go down to the liver and the colon. And when we see the bacteria eating fructose, we know that person needs to cut down on fruit in order to prevent fatty liver. We can see if you're eating too much fat for your body. Again, in this era of paleo and ketogenic diets, this is becoming a problem for some people. When we eat fat, we produce bile in order to absorb that fat. And this bile changes the environment in the intestine, and it promotes many harmful bile tolerant organisms and causes them to grow. We can also see the microbial pathways for fat metabolism and whether they're resulting in beneficial or harmful products. Many of the microbial metabolites from a high fat diet can actually lead to insulin resistance. We can also identify those people who will benefit from eating foods like pomegranates, walnuts, and raspberries, since they need the bacteria that are capable of converting the ellagic acid in these foods into the anti-inflammatory urolithin A compound. Now, if they don't have the bacteria that are capable of doing this in their microbiomes, perhaps they should focus on other foods that will provide more benefit. And this is an example of a publication that shows that. Oxalate containing foods such as spinach can be harmful unless you have the microbes present that can metabolize it into a non-harmful substance. We can easily see whether these microbes are present and whether you should eat maybe a little less spinach, bran, beets, and rhubarb. Now, we've been told that resistant starch is good since it's a food for bacteria that can convert into that beneficial short chain fatty acid we saw earlier called butyrate. However, I'm sure you know by now, some people have the bacteria and pathways that are very good at using these starches to make butyrate, and some of us unfortunately don't. Other foods such as garlic and onions have other fibers that also feed certain bacteria. But if the wrong microbes start to ferment these fibers, it can result in severe gastrointestinal symptoms. We can also see the bacteria and pathways that utilize the EGCG in green tea to produce beneficial and sometimes harmful substances. We don't want to make everyone drink lots of green tea just because it benefits some people. And the porphyries in seaweed can also be metabolized by specific bacteria. And it makes sense that these types of bacteria are most prevalent in Japanese microbiomes. The diazidin in soy is again metabolized by specific bacteria to produce equal, which is a selective estrogen receptor modulator, which is a beneficial effect. Now, not everyone has the bacteria capable of doing this, so soy will not benefit all women. Now, certain minerals like iron in foods, when they're combined with an inflammatory microbial environment, they can promote the growth of pathogens and turn these potentially pathogenic organisms into highly pathogenic ones that can lead to severe disease. It's good to know when to cut back on iron intake before it causes big problems. And salt can be beneficial, it can be neutral, or it can be harmful to specific people. It can cause significant inflammation by stimulating Th17 cells. We can see if salt is causing this in you and whether you need to cut back on salt. Even so-called good salt, such as Celtic sea salt or Himalayan salt, can have this effect. One of the ways our bacteria can actually help us deal with inflammation and oxidation is by using glutathione. We've been able to identify the glutathione pathway in the bacteria of many of the samples that we've looked at. And we can see very precisely whether someone is taking glutathione or if their bacteria are making it because they're dealing with a toxic or inflammatory environment. And bacteria are one of our main users of glutathione. In fact, our microbiome, not our liver, is really our biggest detox organ. We use bacteria to clean up oil spills, so it makes sense that they can help us detoxify. Tryptophan is an amino acid that we get from foods and supplements, and it can get converted by our bacteria into many different types of compounds. Some of these, like IPA, are neuroprotective and can protect the brain against diseases like Alzheimer's, while others are a little bit more detrimental, such as indoxyl sulfate. This metabolite can contribute to oxidative stress in the endothelial cells, the cells that are lining our blood vessels, and it can lead to heart disease. So, when we see certain tryptophan pathways elevated, we can make recommendations such as specific probiotics or prebiotic foods to push it in a more beneficial direction. Our bacteria are also capable of making amino acids from certain sugars and starches. We often see this in people who consume a high vegetable diet. Many of these amino acids made by the microbes are absorbed and used by our bodies. In some cases, these can be very beneficial, and in other cases, it can potentially lead to things like insulin resistance. So, the more we delve into what our microbes do and are capable of doing, the more amazed I really am. Our microbes clearly metabolize things like our hormones into potentially good or bad ones, but they can also make hormones such as estrogen and testosterone. And we can see when our bacteria are contributing to the reabsorption of toxins, of drugs, and hormones that our liver has really worked hard to get rid of. We can also see whether you're likely doing high intensity exercise by looking at how well your microbes are producing energy. And we can see whether you're expressing any microbial enzymes that help your body extract energy more efficiently from your food. Depending on your activity level, this might be a good function or it might not. Now, this is really exciting. We finally have the tools to know what to do, with the ultimate goal for someone like Joe to know what will be optimal for him so he can achieve his goals. You can see that precision wellness is the future of nutrition. It's the difference between Joe just following the latest fad diet and getting worse, or knowing precisely what he needs to eat in order to lose weight and get rid of inflammation and be truly healthy. At Viome, we've created an AI-powered wellness companion delivered through an easy app, so this technology is available for everyone. Thanks for watching, and please let us know if you have any questions.